So with that, I think it's appropriate right now that I take a few moments to introduce our first keynote speaker, Professor Tom Wilkinson. He is a professor of respiratory medicine at the University of Southampton in the UK in the Faculty of Medicine, and an honorary consultant at the University Hospitals Southampton. He trained at Cambridge University in UK and also at Barts and the London School of Medicine, and completed his PhD at UCL, studying disease mechanisms driving infective exacerbations of COPD. He is lead of the Southampton COPD Research Group and Airways Disease Research Theme Lead for the NIHR Biomedical Research Center in Nutrition and Respiratory Medicine. His research seeks to improve our understanding of those mechanisms which drive susceptibility to respiratory infections and exacerbations in those patients with chronic lung disease, and to develop new vaccines and therapies to have an impact on these areas. Tom has taken these mechanistic discoveries from basic science to translation into new treatments, and we're going to hear about one of those today. He was the co-chair of the British Thoracic Society Home Oxygen Guidelines Standards of Care Committee and contributed to the UK National Nutritional Guidance for COPD. He's an associate editor for the journal Thorax and is co-founder of the health technology company MyMHealth. He's published more than 100 peer-reviewed publications in scientific journal and reviews on the topics of airway disease, exacerbations, and airway immunology. His work has been recognized by national and international awards, including the Maurizio Vignola Award for Innovation in Respiratory Medicine in Europe by the ERS. We're very fortunate to have Dr. Wilkinson join us this morning, and welcome, and I invite you to start your presentation straight away, Tom. Thank you.
Splendid. Well, thank you very much for the introduction. Just let me get set up with slides and we'll get going. I just hope I can change my slides. So I'm a physician. This is a conflict of interest statement which we're obliged to just present. We're funded broadly by industry and we work closely to develop new drugs with pharmaceutical companies. This is us. So I'm based in Southampton in southern England, where we have a rainy, cold day at the moment. I work with an exciting team of young scientists, so doctors and nurses and scientists working closely together to try and take our understanding of disease from the clinic into the laboratory to work out mechanism, and then back into the clinic with new therapeutic ideas. And I've been working in the area of respiratory infection, particularly viral and bacterial infections in the lung for the last 20 years. And really, we've pivoted our entire program of work into COVID in the last 18 months, really because we saw this as the enormous problem that it became quite early on in this cycle of the pandemic. And I'd like to share with you some of those experiences. So this is the current state of the art in terms of treatment for COVID-19. We have really no definitively effective treatments. And these are the drug study results from the recovery study in the UK, which is one of the largest trial platforms for COVID therapies. In terms of the state of the art now, we're giving patients in the UK dexamethasone, the steroid, and we're giving them tocilizumab, the anti-IL-6. There's a role for remdesivir, the antiviral, but the standard of care is not adequate to save many patients' lives once they're admitted to hospital. And you can see from these graphs that the mortality rate for the severely unwell remains high in the UK and globally at over 30%. So there needs to be a continued effort now, even as the vaccines are rolling out, to discover new therapies. So we would like to focus the next few minutes really on thinking about approaches to improve outcomes from viral infection, and particularly COVID-19, and to think about particularly the innate immune response and focus on interferon therapy. Here's a schematic of the timeline of the immune response to any viral infection. And as you'll be aware, the initial response, the innate response, rises very rapidly and is very important in control of the initial response to any viral infection. This is particularly important in viruses which are novel pathogens, which the immune system has not seen before, because we don't have an adaptive response to the SARS-CoV-2 virus that is widely held in the population prior to the pandemic hitting. So there's an importance to understand how we could augment an innate response, which may be able to abrogate the severity of this disease. And this is where we're going to focus. So we're going to think about interferon beta. So interferon beta is, of course, a naturally occurring protein which orchestrates these initial innate defenses. It's potentially a pan-viral because it works not just for one virus, but by switching on this host defense, could work against any particular virus. It's delivered in our studies as a local delivery using an inhaled therapy, and we'll think about why that may be useful. It increases the bioavailability of the drug at the site of infection in the airway, and it's been formulated by a spin-out company from the University of Southampton to particularly be well tolerated in the lung with no incipients that can irritate the airway, and it's delivered in a very fine mist so it can get right down into the lungs. And we've been working to develop this therapy many years before COVID hit in order to try and treat exacerbations of asthma and COPD, which are often virally triggered. But when we started to see the science emerging around COVID-19, we became aware that there may be an actually particular role for interferon therapy in the context of this syndrome. This is one paper which identifies that potential because it shows that in patients with severe COVID, there's actually a deficiency in interferon responses, which is unusual in the context of a severe viral infection. And these data show that patients who end up in intensive care have a suppressed interferon response, but a heightened inflammatory response as signified by levels of IL-6. And so that we have an interesting syndrome here where we need to understand this phenomenon and to potentially augment the interferon response if possible. So there are a number of other emerging parts of the evidence base which point to an important dysregulation of interferon in COVID-19. So SARS-CoV-2 itself is a virus which can disable interferon responses, suppressing interferon production through the open reading frame proteins and others, perhaps more so than other respiratory viruses. We know that patients with a genetic defect which impairs interferon production have really poor outcomes from COVID, and that certain patients in this recent study in Science actually manifest autoantibodies against interferons, and that's associated with poor outcome. We know that a particular population are vulnerable, the aged population, have poor innate defenses and produce lower amounts of interferon than others. And there's also key mechanisms in other at-risk groups, such as diabetics and obese patients, which again subvert the interferon response. So all this evidence is pointing in the right direction that this treatment may offer some opportunity to improve outcomes in COVID. And as I said before, we'd worked significantly with vulnerable patient groups, particularly asthma and COPD patients, using this drug in order to try and dampen down the inflammatory response to viruses at exacerbations of these conditions. And we'd shown the drug to be very well tolerated in this vulnerable patient population. When you're giving immunoactive drugs into the lung, obviously there can be potential downsides to this, inflammatory stimuli, cytokine storm production, and we'd shown quite definitively that we could engage the interferon signaling pathways without any of these poor downside sequelae. So there are also a complex network of interferons and other innate proteins. So why select interferon beta? There is only one interferon beta, but over 10 types, 13 types of interferon alpha, and they all have a slightly different immunomodulatory effect from each other. We know that both interferon alpha and beta can suppress viral replication, and studies obviously have been more well thought out in flu and in other viruses prior to COVID. But we know that interferon beta, at least in vitro, may be a more potent inhibitor of SARS-CoV-2 infection, and that the antibodies that we do see emerging, as mentioned from the Science paper, are more directed against interferon alpha than beta. So there is a rationale there that beta may be an optimal choice of therapeutic. We also know that in vitro, interferon beta can suppress the COVID coronaviruses, so MERS, the previous coronavirus, and indeed now recently SARS-CoV-2 has been shown in vitro experiments to be heavily suppressed with augmented interferon in laboratory experiments. And we were just starting a new program, a large-scale program, this time last year in January, February, it was just about to round up when the COVID pandemic hit the UK. And what we actually did was we picked up this clinical trial in COPD and reworked it to face the pandemic. So we were looking at giving inhaled interferon via nebulized form into a trial design where we use a platform which can rapidly diagnose the virus. So these are ubiquitous now, but a year ago they weren't. And we started to think about designing this study for the new disease, which we had little experience of at that time. So here it is. This is the Synergen 016 trial. This is a trial that is completed, and I'll show you the data from it. This is a randomized, double-blind, placebo-controlled trial of interferon beta, and the aim of the study was to determine in a hospitalized patient population if we could improve outcome using a WHO ordinal scale. So it's a clinical measure of how well or unwell patients are. I'll take you through that. We had a pre-planned analysis to complete 100 patients randomized 1-to-1, so 50 and 50 in placebo and interferon beta arms. And we were dosing these patients using that nebulizer device once a day during their hospital stay and to continue at home if they were discharged prior to the 14-day window. And here's the WHO scale. So when you're trying to treat a new disease like COVID-19, it's very hard to design clinical trials because a year ago, we knew very little about the natural history of this condition. We had reports from China explaining the potential severity of the disease. And we knew that a lot of patients were developing respiratory failure and ending up in ITU. The WHO did a stellar job really to pull together this scale to characterize in a fairly objective way the stages of severity based on the clinical status of the individual patient, and to help clinical trialists compare one drug to another using this same scale to determine the impact of the treatment. So it runs from 0 when you're well with no symptoms to 8 at death, with every stage in the usual care pathway between. So we had a fairly open inclusion criteria for the trial. We looked at including patients admitted to hospital who were over the age of 18, who had a positive test for SARS-CoV-2 PCR, but not yet on a ventilator. So it was important that we were giving them this drug through an oral inhalation route, so they weren't yet in the ITU setting. And we followed these patients through the trial structure. We worked in 9 hospitals in the UK, and we got the study up and running within about 2 weeks of getting going with coronavirus research in the UK. If anyone's done clinical trials, usually it takes months or even years to get moving. So things got going very quickly. We randomized 101 patients, and you can see here we had some withdrawals from each side. People were very sick, and we had 3 deaths in the placebo arm, but none in the treatment arm. And we'll look through the results now. So here's the sort of patients that we had included in the trial. Patients were middle-aged. There were more men than women. There were a mixed population ethnically with a predominant white population, which is descriptive of the patient group that was being admitted at the time. There were comorbidities in both arms, and we know that severity of disease tracks with comorbidity, so it's important that we were reasonably well matched for those. But patients had other issues as well, ongoing frailty, and we know again there's a risk factor there. So we collected all those data. So the results came in, and when you've done clinical trials, you know you put an awful lot of work and sweat and tears into them, and then you get the results through, and you're never sure what to expect. We knew the drug was safe. We had interim safety reviews throughout the program, but we didn't know what the efficacy signal would look like. And so we were very pleased to see a positive signal from this study. The odds of improving on the treatment compared to placebo were significant statistically and clinically. So you're over twice as likely to get better on the drug than on the placebo arm. The odds of deteriorating, so moving from the hospital admission down into ITU or worse, were a lot lower on the drug than placebo. So that was a 72% reduction in that risk, although it wasn't quite significant in a small study. And as I said, 3 patients died on placebo with no deaths on the active arm. So all very encouraging. If we look at those data graphically, we can see that everything is to the right side of that line. So when you do small studies, early phase studies, when you're focusing initially on safety, you need to look at the entirety of the data, and one p-value here or there is not necessarily telling us what it would do in a very large trial. And this tells us that actually all the outcomes we looked at using the Oski scale were very much in favor of the drug, and that some of them were significant, many of them statistically significant, and that this would be a clinically meaningful result if we could replicate it in a larger trial. So we also looked at not only what happened in hospital, but how quickly it took you to get better and back on your feet at home. And we know now there's an emerging narrative in COVID that this is not just an acute illness, but there's a long tail to this illness for many people. And so understanding how long it takes to recover is also an important signal. So here is the data. So these are the percentage recovery rates in placebo, in the open circles, and Synergen drug in the blue, the interferon beta. We can see that these separate quite early on, within about 3 days of dosing starting, and remain separated with a statistically significant difference in recovery rates at day 14 at completion of treatment, and an even greater difference at day 28. So you're twice as likely to make a full recovery with no symptoms at all if you're on the drug than on placebo. And again, we looked at time to hospital discharge. So this is the amount of time spent in hospital. There was a signal here, not statistically significant, but it looked like patients were more likely to be discharged early on the drug than on placebo. And that's an important statement if you think at how busy hospitals are and how resource poor many healthcare settings are. Being able to get people out of hospital early would be an important outcome. And so what about symptoms and how the patients feel? So in COVID, we know that there's a conglomeration of symptoms that patients talk about and experience. So of those, respiratory symptoms are paramount. So here we're looking at cough, we're looking at breathlessness, we're looking at sputum production, but also there are symptoms around fever, muscle ache, fatigue, headache, and indeed depression. So this is focusing on the respiratory symptomology. And what this shows us is that there was a gradual improvement in symptoms over the duration of the trial. And we used a score here called the BCSS score, which is a respiratory score. Had been no validated COVID scoring systems at the time. So we borrowed a score from our previous respiratory studies, and we used this in all patients. And this shows us as over the 14-day window of the treatment that patients do get better. but noticeably the breathlessness improved significantly quicker in the Synergen active treatment arm compared to placebo. So patients were self-reporting that they were feeling less breathless if they received the drug, and by the end of the trial, a majority of patients had actually improved significantly in terms of the level of symptomology, which is an important finding considering the length of symptoms in many of these patients. And then we looked at the treatment adverse events. So when you're delivering a phase 2 study, it's absolutely vital that you can demonstrate that your drug is safe as well as has a signal of efficacy. And so in real time, these safety data are captured and reported, and they are looked at by an independent safety review group and then by the complete trial team. And what we saw was actually quite a matched pattern of adverse events. So adverse events are things that the clinician reports on behalf of the subject in the trial. And these are things like cough, a headache, other symptoms that may be associated either with COVID or indeed with a drug side effect, as listed here at the bottom of the slide. And we saw that they were quite well matched. There were actually more serious adverse events in the placebo arm than in the Synergen arm. Overall, there was no real signal of adverse events associated with the therapy itself. And so we looked then beyond the primary outcomes to a range of secondary outcomes to really explore where this drug was having its biggest effect. And I won't expect you to read through this in detail. There's a lot of numbers on that slide. But the important point was that everything was heading in the right direction. So wherever we looked in this trial, we saw a signal of potential benefit. And that's really quite an unusual situation, having done drug development for many years. And so we were really keen to understand what sort of population perhaps that we were seeing the strongest benefit in. We've seen in the past that the dexamethasone study, which I reported on at the beginning, had shown a benefit in some patients, but not others. So dexamethasone is indicated in patients on oxygen therapy or intensive care, but not in patients with milder disease, pre-hospital, or indeed in hospital not requiring oxygen. So we were interested, even in our very small trial, to see if we could see any compartmental responses. And so we looked at a number of outcomes, including patients with more severe disease and how they differed from patients with milder disease. And patients with more severe disease seem to have even stronger response, so an odds ratio of over 3 in terms of recovery compared to the overall population. So what does this mean? It means that there is a clear indication for this therapy, if this is repeated data in larger studies across the disease spectrum, but there may be actually an augmented response in even more severe disease than the average hospitalized patient. And that's relevant because I think there was a lot of anxiety prior to these trials about giving inhaled interferon to patients with severe disease, because it could tip over a patient into a more inflamed status, was the hypothesis, but we didn't see that, and I think that's a key finding. Okay, so in summary then, this trial of hospitalized COVID patients with interferon beta has shown increased odds of improving clinical status, increased the likelihood of recovery, and significantly reduced breathlessness during the treatment window. It was well tolerated, and we feel that this is a promising early phase trial. This isn't the whole story, and I'll take you through where we're headed next. There's a community study, which I'll tell you about. So we're very interested in exploring this disease in the pre-hospital patient population. Most patients, thankfully, don't end up in hospital. So we need to see if a treatment could actually prevent them getting into hospital, improving their recovery rates. And there's a phase 3 study, a much larger study that's now underway, and I'll tell you a little bit about that. So what about the community setting? So one of the challenges of doing trials in COVID out of hospital is the risks of infection and the lockdown effect on ability of patients to move around. And the conventional approach to actually studying drug effects by asking patients to come into hospital for visits was no longer feasible. So we worked with the University of Southampton. The lead on this project was Dr Francis, who's a GP who works in, a professor of primary care who works in the community setting. And they developed a very ingenious way of actually completely delivering a clinical trial remotely without any direct face-to-face interaction with the patient, a bit like this conference. So patients were recruited using a web site, and then they went through a screening program. They were once initially screened, they were consented by an expert a nurse or doctor, and they had the whole usual visit and screening process performed remotely. And if they passed the screening process and they had a positive COVID result from their local testing, then the drug was expedited right to their door. So it's delivered to their door. They were then shown using a video link how to use the nebulizer, and then they went into their usual treatment plan at home. So really quite an ingenious way of delivering a trial. And 120 patients have gone through this study now. It completed recruitment in January, and we'll get results from this trial within the next few weeks, which will tell us how effective this drug is in the pre-hospital population. And now, as I said, this is a journey which is only half finished. So the phase 2 study gives us a very positive readout, but it doesn't give us a large population of patients that have been dosed. So we need to be sure, A, that in a large study that we confirm these results, that B, there are different and diverse populations that we need to treat, the older, the younger, different ethnic groups and in different hospital settings, and the standard of care is evolved. So we need to be sure that this drug will be effective across all the current standards of care that are offered. So this is where the Sprinter study comes in. This is a phase 3 trial of the same drug, but with a different device now. So we've moved from the small iNeb nebulizer to this device, the Aerojet nebulizer, which can give the drug quite straightforwardly into the lung. It's and again a mesh nebulizer, but it can run in parallel with different circuitry in hospital settings. It's quite easy to administer, it's cheaper, and it's scalable, so we can produce many more of these devices. And this is a randomized study of 600 patients now in the pre-ICU state, and we'll be looking at time to recovery again, how quickly we can get patients out of hospital and how quickly they can become better again, and prevent progression to severe disease, look at the death signal, and look at the prevention of long COVID. And this is a study that's up and running in the UK now in over 50 sites, and in over 15 sites in the UK, but we'll be running this trial globally. So many of the countries that have dialed in today will have sites of this therapy. So you may be able to track this locally. And we're looking at including over 60 countries and delivering this trial over the next few months. And then if this was a positive study at that stage, we'll be looking for licensure to use in this disease. There's also another study ongoing. So this has really sort of caught the imagination. You'll be aware that the Active-2 program is a US funded trials program, and that the interferon therapy has been adopted into this program. It is a community-based trial where we're looking initially at a small population and then selecting the appropriate endpoints, and if safety signal is maintained, expanding that rapidly into several thousand patients to show definitively that this could be a potential therapy or not in the pre-hospital setting. So this has all happened very rapidly, and that's quite an unusual picture for drug development. So just to rehearse that timeline really, so we heard about the coronavirus outbreak in early January in our department, and we were talking to colleagues both in China and then in Italy, which was the first country really to be affected in Europe back in January last year. By February, we had seen evidence of transmission into the UK, and we decided to pivot our entire hospital program and move the trial from COPD into interferon beta. By the end of February, we'd written the trial's protocol. We'd start to sort permissions from the regulators to start the trial. And in early March, we actually submitted the program and received approvals within a week or two, which usually it takes months. By the end of March, we'd recruited the first patient, and by end of May, we'd completed the 100-patient study, and then we released the results in July. And then we had to go into fast scale up. So one of the challenges when you're doing drug development is not just the trial itself, but it's the production of the medication and then of the devices. So the company, which is a small company, has moved very rapidly to actually scale up drug manufacture and work with international regulators to seek approval, both for the larger scale studies and indeed for long-term use if we're successful. So this has been an enormous collaborative effort, and I'm coming to the end of my talk now, so we'll have some time for questions. This is really done in a new way. So COVID has done an amazing thing, I think. It's broken down barriers which previously existed. So we link now on Teams calls nationally and internationally routinely. We work across previous silos, and this study is really testament to that. We've had an international group of scientific advisors. We've worked closely across NHS, that's our health service, and university boundaries in the UK. We've worked with a range of different trials, groups and hospitals. We've had support from the National Institute of Health Research in the UK and the infrastructure to deliver studies. And we've worked with industry, with Synergen to produce the drug, with the Aerogen and iNeb nebulizer companies. And, you know, it's a new way of working, fleet of foot and task orientated. And really, it's a very exciting time, I think, coming out of COVID now to think we could continue to work in a agile way to solve many of the health problems beyond the pandemic, which we've struggled really to tackle in the past. And respiratory infections are certainly part of that story. And we hope that many of the lessons learned can be carried on into the future. So I'll stop there. I daren't touch my screen in case I do anything I shouldn't. But thank you very much for your attention, and I'll wait to instructions as to how we're going to do the questions.
Hi, Dr. Wilkinson. Thank you for that amazing presentation. So we do have quite a few questions coming in. I will start by reading out the first one. So what are the advantages and disadvantages of using inhaled interferon beta treatment when compared to other treatment for SARS-CoV-2 infection?
Good question. So I think we don't know all the answers yet, but certainly the concept here is that we're giving a bioactive compound, interferon beta, directly into the airway. So we're using a nebulized formulation, which creates a fine mist, and the droplets carry the drug deep down into the lung, where we feel that the active virus is replicating. The reason for doing this is we're able to give a much higher local dose, so the bioactivity in the lung is much greater using the nebulized formulation than were we to use an injected form. If you use an injected form, the drug is distributed across the whole body, and so you'd have to use a very, very high dose with the problems that would cause to get the same dose into the lung itself. So there's the advantage of using an inhaled formulation. And we also know that the virus, that this is pretty potent stuff. So interferon beta naturally occurring switches off the entire virus machinery, viral replication machinery, when we study it in the laboratory. And that's different from other disease drug mechanisms. The challenge is just that, is getting this drug nebulized and into the lungs of patients. Now, some of the patients are very unwell, so they may be on a high flow oxygen system, or they may be ventilated. And it's much more difficult to distribute the drug using those ventilated supported patients because there's a dead space issue and the drug is not necessarily deposited in the same way. So the team has been working really hard to understand how this approach could be used in more severely unwell patients. And we've made significant progress with that. And I think within the next few months, we'll be launching another trial phase in the ITU setting, where we may be able to offer those patients the treatment as well.
Thank you. And then a similar question that can kind of lead off of that is, someone is interested to know whether the drug was used alone or as a combination therapy with other COVID-19 protocol medications.
So we used the drug with the usual standard of care. So standard of care has evolved over the last year or so, but we felt it was important that the drug was shown to be beneficial over and above what the current standard of care was. So that would have been steroids early on in the pandemic phase, and more recently it will be with the anti-IL-6 therapies, which are now licensed for routine use, remdesivir. The issue is that none of the current standard of care patient therapies are that effective. If you put them all together, there is a clear benefit, but it's not definitive. So we feel that we have to lift the bar even further, and so interferon beta is not being used instead of, but on top of those treatments.
Thank you. And then another question we have is, any role or attempts of using interferon for latent or primary tuberculosis?
Oh, good question. So we haven't looked in TB, and obviously there's potential use for this drug in a range of infectious diseases. So we've looked at respiratory viruses, but the innate response is important, against a range of pathogens, of which TB may be one. We haven't done that work, but I think it's a concept that we'll need to take forward in terms of chronic lung infection after the pandemic and see if there could be potential benefits outside. It'd be good to look at some in vitro data on that. TB is not my particular area of expertise, but it's an interesting concept. Thank you for the question.
And then another question is, what age group drug trial was tested, and was there a difference in the drug efficacy or recovery rate with different ethnic backgrounds?
So, good question. So we took all adult admittees over the age of 18. So the average age of a hospitalised patient in the UK at the moment is in the late 60s. It's changing because of the vaccines have been impacted on the more elderly. But our patients in the first study were in their late 50s. So this is typical of an early phase study where you don't tend to use the drug in the most vulnerable population. Moving on, the drug will be given to everyone in the larger phase 3 study. And what we would imagine, in fact, is that the more elderly you are, the more there is for potential benefit, because your own innate production of interferon beta is embarrassed as you age and the lungs become more senescent in terms of an immune response. So I would postulate that there may be additional benefit in the elderly. In terms of the ethnic groups, we weren't really powered to look at differences between groups. We know that certain ethnic groups seem to do less well in terms of their response to COVID, and I think there's a lot of interest as to why that might be. Again, there is a potential to understand that augmenting the innate response using interferon beta may actually benefit some of these more vulnerable populations. But again, we'll have more data. Certainly in the ACTIV-2 study, we should have thousands of patients in the trial, and we'll be able to look at the different ethnic populations and their potential response.
So you just mentioned that this would be effective in an older population. One of the questions is, would the SNG1, could it be used preventatively in an older population?
Yeah, so this is I think a really interesting question. So we have particular vulnerable populations, particularly in care homes and in nursing home facilities, who are incredibly vulnerable and almost trapped. So if the virus transmits into that environment, those patients have a terrible outcome. We saw that in the first wave in the UK with mortality rates in those settings over 80%. So I think there's a real study to do here, and actually Dr. Fish and I have spoken about this. Could we give a prophylactic dose to people that are at extremely high risk, potentially prevent severe illness? And I think that's a study that needs to be done. I think there are studies of that nature going on with other treatments, and I'd very much like to explore that approach.
Maybe I can jump in here then with a couple of questions, Tom.
Sure.
Thank you. That was an awesome presentation. Leah, unfortunately, I can't get on the Q&A. That's why I'm having to jump in here. So have you any data, or are you planning to have any data on the rate of viral clearance?
So we've got scant data on that. I think we'd love to have better data, to be honest, but the initial pandemic was a foot race, and hospitals were just chaotic, and we had to negotiate sort of the burden of sampling that hospitals felt that they could manage, and repeated swabbing and even saliva testing, which now would appear to be a sensible approach, weren't designed into the initial trial. Moving on, we will be doing nested studies where we're looking at repeated measurements, and we would expect to see, one would imagine, certainly in a saliva sample, a reduction in viral carriage. But the dynamics of that are uncertain at the moment, and in a few months' time we'll have better data on that.
And my second question is, do you think that based on the fact that you're using an inhaled interferon, this is why your data are so much more compelling and positive than the studies where sub-Q or IM in administration has been looked at in a number of different studies?
Yeah, it's slightly head scratching, isn't it, that we've seen various different outcomes using interferons, alpha, beta, injected, and now inhaled. I think that bioavailability, in the lung at the site of infection is important. There may be issues around injecting different formulations of the drug in different clinical settings. Many of the interferon studies have been small or didn't have a control arm, so it's always difficult to interpret those sort of data. I think there is a biologically measurable difference of using a high dose inhaled form than a comparable injected form. And obviously the proof will be in the larger trial result, as we know that injected interferon in larger studies has not been that useful. So fingers crossed, and we hope we see a positive result soon.
All right, thanks very much. Okay, Leah, I'm done.
Thank you very much for answering all those questions. You'll still be able to type answers in the Q&A function if people still have questions rolling in. So thank you very much for that presentation. And
thank you so much, everybody. And sorry about the slide show earlier on. Appreciate it.
It was a perfect presentation. Thank you.
Thank you.